classification, the term algae means a group of artificially assembled organisms including both prokaryotic and eukaryotic members. The cyanobacteria, previously known as cyanophyceae and commonly known as blue-green algae, are composed of prokaryotic cells as they are prokaryotic in nature. On the other hand, rest of the algal members are eukaryotes, and obviously their plant body is composed of eukaryotic algal cells. Now, the prokaryotic algal cells or the cyanobacterial cells are comparatively smaller in size, and these cells are generally encircled by a gelatinous sheath material, and there is prominent cell wall. Inside the cell wall, there is no membrane bound cell organelles like nucleus, mitochondria, Golgi bodies, etc. Only the membrane bound structures are the sac-like thylakoids, which are present at the peripheral zone of the protoplasm and are known as the chromoplasm. These thylakoids are actually the pigment bearing structures. At the central region of the protoplasm, which is known as centroplasm, the naked DNA is present, which is the genomic material of cyanobacterial cell. Besides this DNA material, there are few granules in cyanobacterial cells like polyhedral carboxysomes, which is the site of carboxylase, and the cyanophycin granules, which are proteinaceous granules. And a large number of vacuoles are present inside the cyanobacterial cell, which give the cells buoyancy. Now, in contrast to the cyanobacterial cell, all the other members are composed of eukaryotic cells. Now, a typical eukaryotic algal cells are just like a typical eukaryotic cells, having a prominent cell wall, membrane bound nucleus, and other cell organelles like mitochondria, Golgi bodies, and others. Besides this, a specialized chloroplast is present in almost all eukaryotic algal cells, and there is presence of flagella either in vegetative cell or in reproductive cells of algae. The dinoflagellates and the euglenoids are made up of mesokaryotic cells, where we can see only the condensed chromosome throughout their life cycle, though there is no well-developed spindle formation. Now, if we consider about the extracellular coverings of algal cell, then we can mention three structures. These are sheath, frustules, and scales. These sheaths are the polysaccharidic structures which are present in almost all algal cells, especially in cyanobacterial cells. Sheaths are the extracellular gelatinous covering of algal cell, specially present in cyanobacterial cell. These sheaths are polysaccharidic in nature and are composed of a number of monosaccharides like glucose, galactose, mannose, rhamnose, arabinose, and uronic acids, glaucuronic acids, and galacturonic acids. Presence or absence of sheath in cyanobacterial genera sometimes are very important because they are considered for identification of the taxa. Like in Oscillatoria, sheath are absent, but in Lyngbia, a prominent sheath is present. But both the genera are identical in morphology. Therefore, here sheaths are essential for identifying the taxa. The sheath contains the water, and they actually protect the cyanobacterial cell against desiccation. Amount of sheath development depends upon the habitat of the cyanobacterial cell. When the cyanobacterial cell grow in aquatic habitat, sheath material is much less, but when they grow in terrestrial environment, sheath is comparatively more. Now, sheath are generally colorless, but sometimes sheaths may be colored. It may grow in acidic soil when the sheath is red, or when the cyanobacteria grow in basic soils, sheaths are blue. Now, in the genus Cytonema, the sheath is yellow due to presence of cytonemin pigment, which protects the cyanobacterial cell against UV radiation. Now, frustule is the extracellular siliceous covering of diatoms or the bacillariophycean members. These frustules are actually the box-like structures encircle the diatom cells. They have two parts. One is upper lid-like structure or known as epivalve or epitheca, and the other is the lower half, which is known as hypovalve or hypotheca. So, we can see the diatom frustules from different angles. When you see the diatom from the top, then it is called the top view. When we see the diatom box from the side, it is called the girdle view. Now, these diatom frustules are provided with many minute pores which are known as puncti. These puncti are arranged in definite rows which are known as striae. Now, these striae or the arrangement of puncti are very important because they help to identify the diatom taxon. These striae, when arranged in radially, then it is called the centric diatom. When these striae are arranged in reference to a central line or raphe, then it is called the pennate diatom. Pennate diatoms are generally the box-like. Now, these diatom frustules, as they are made up of silica, they remain in the seabed after the death of the diatom frustules. And in course of time, these assemblage of diatom frustules are fossilized to form the diatomaceous earth. And these diatomaceous earth are very economically important. They can be used for filtering, they can be used for production of soundproof materials, or in different industry these diatomaceous earth are present. Now, scales are another extracellular coverings present in different groups of algae, especially in chlorophyta, haptophyta and dinophyta. In chlorophyta and haptophyta, the scales are organic in nature and they are made up of polysaccharides, whereas in haptophyta the scales are made up of this calcium carbonate and it is known as coccoliths. Cell wall is one of the prominent structure of algal cell and it varies with different groups. In cyanobacteria, the cell wall is made up of murin or peptidoglycan. The murin is actually the alternate unit of N-acetyl muramic acid and N-acetyl glucosamine. And these polysaccharides are joined together with 4 amino acids: alpha-alanine, beta-alanine, glutamic acid and diaminopimelic acid. And this diaminopimelic acid is the characteristic feature of the prokaryotic algal cell. Now, on the other hand, the eukaryotic algal cells are composed of two components. One is amorphous components and other is the fibrillar component. The fibrillar component is mainly composed of cellulose, which is the 1,4 linked beta D glucose, and the glucose sometimes is replaced by mannose, xylose or chitin. In some algae, calcium carbonate deposition is also there on the cell wall and they turn the algae into the calcareous form. Now, in 70% of the rhodophycean members, the amorphous region is made up of agar-agar or carrageenan. This agar-agar is actually composed of agarose and agaropectin. It is the sulfated glucans. This agar-agar is very much economically important. They are used in different food industry, medicinal industry and are also used in laboratory purpose for growing other microorganisms. Then now in the members of Phaeophyceae, the cell wall is mostly composed of the alginic acid, which is actually 1,4 linked glaucuronic acid and manuronic acid. This alginic acid is again with of industrial importance. They are used in different industry including the paint industry and other food industry. Nucleus is one of the important structure present in all algal cell except the cyanobacteria. The cyanobacterial cell contains the naked DNA without histone protein at the centroplasm area. The molecular weight of this genomic DNA is 1.6 to 8.6 into 10 to the power 9 Dalton. On the other hand, the eukaryotic algal nuclei are just like general eukaryotic nuclei with nuclear membrane, pores and the chromatin matters. Only in dinoflagellates and euglenoids, a different type of nuclei are present which are known as mesokaryotic nuclei, where the chromosome remain condensed throughout their life cycle and there is no spindle formation. It was believed that these mesokaryotic nuclei are in between the prokaryotic nuclei and the eukaryotic nuclei, but in recent studies it has been found that these mesokaryotic nuclei are actually the degenerated eukaryotic nuclei. Now, one of the most important structure of algal cell is their chloroplast. Chloroplasts are specialized structures and present in very well organized manner in eukaryotic algal cell. Now, these chloroplasts are actually the pigment bearing structures. In cyanobacteria, chloroplasts are absent and there the thylakoids are present at the peripheral region of the protoplasm. These thylakoids remain free or in grouped and embedded in stroma or ground matrix. The stroma contains 70S ribosomes and the DNA. These thylakoids are actually encircled by the membrane to form the chloroplast. Now, the chloroplasts are present in different forms in different groups of algae. Ultrastructurally, it has been found that chloroplasts are double membraned in Glaucophyta, Rhodophyta and Chlorophyta. In Rhodophyta and Glaucophyta, the thylakoids remain free in the stroma, whereas in Chlorophyta, the thylakoids are grouped together to form the grana and are embedded in the stroma. In eukaryotic algal chloroplast, some pyrenoid or specialized structures are present which are actually the site of starch synthesis. Now, on the thylakoid surface of different groups like Cyanophyta, Rhodophyta and Cryptophyta, some macromolecular structures are present which are known as the phycobilisomes. These phycobilisomes have two parts. One is chromophore parts and other is protein part. These chromophore parts actually contain the blue-green or the red pigment phycocyanin, phycoerythrin and allophycocyanin. This pigment part or chromophore part is attached with the protein part with linker protein. Ultrastructural studies of chloroplast is very important on phylogenetic point of view. It has been found that in some groups these chloroplasts are two membraned, in some divisions chloroplasts are three membraned and in other it is four membraned. In two membrane structures, the chloroplasts are present in Chlorophyta, Rhodophyta and Glaucophyta, and three membraned chloroplast structures are present in Euglenophyta and Dinophyta. The outer membrane of these three membraned chloroplast is known as chloroplast endoplasmic reticulum because these membranes are provided with ribosomes. Now, four membraned chloroplast can be seen in Heteroconchophyta, Cryptophyta and Primnesiophyta. Here, two are chloroplastic membranes and the outer two membranes are chloroplastic endoplasmic reticulum. Now, besides these chloroplasts, some specialized structures are also present within algal cells which are vacuoles. Different types of vacuoles are there like contractile vacuoles which are present at the base of the flagella. And these contractile vacuoles absorb water and sometimes they actually expel this water from the algal cell, controlling the osmoregulation property of the algal cell. Specialized vacuoles or pustules are present in dinoflagellate cells. These pustules are almost permanent structures and they are actually linked with the excretion of the cell. Now, in cyanophycean members, the vacuoles are actually honeycomb-like structures and they give buoyancy to the cell. These vacuoles get compressed when the cells are with high turgor pressure. Some ejectile organs are present in dinoflagellate cells which actually ejects some microfibrillar structures in response to different stimuli like light, heat and other. Now, most important structure of algae is their flagella. Now, either vegetative or reproductive cells of many algae are provided with flagella. Flagella are absent only in rhodophyceae and cyanophyceae. Now, algae can be classified or can be identified in different groups based on the number of flagella, position of flagella and types of flagella. Generally, flagella are present in the anterior side of the algal cells. When two flagella are of similar size and structures, they are called isocont. Sometimes one flagella is long and another is short surrounding the algal cells, then they are called the anisocont type of flagella. In heterocont type of flagella, the one flagella is fine and is called whiplash type and the other flagella are provided with some hairs and called the tinsel type. Now, in heterocontophyta, the feathers of the flagella again with of complicated types having 3 regions. Now, flagella, I have already told you, it may be the algal cell may be uniflagellated or biflagellated or multiflagellated. In Edogonium, the flagella are arranged in the anterior portion of the male gametes in a ring and is called stephanocont type of flagella. Now, ultrastructural pattern of algal flagella is very important because it is actually used in phylogenetic study. Ultrastructurally, a flagellum has 2 parts. The portion which remained inside the cell is called the basal body and the portion which is present outside the cell is called axoneme. The basal body actually gives the flagella the anchorage, and with this axoneme portion, the motile algal cell or the reproductive cell, they can swim in water very fast. If we consider the cross section of the axoneme part, we will see they are actually composed of microfibrils. These microfibrils are proteinaceous nature and are composed of alpha protein and beta protein. In cross section of flagella, we can see the microfibrils are arranged in 9 doublets at the peripheral region and 2 singlet at the central region. Therefore, the algal flagella also shows the typical 9+2 arrangement. These microtubules are actually attached to each other by protein bands. All these microtubules actually gives the flagella the mechanical strength. Now, there are another specialized structure in motile algal cell, those are flagellar root. From the basal bodies of the flagella, the microtubular roots develop which extends inside the algal cell. These are again microtubular structures and there are 2 types of flagellar root. One is cruciate type and another is unilateral type. In cruciate type, the microtubules are arranged in cruciate patterns and they extends more than half of the algal cells, giving the mechanical strength to the flagella. And in unilateral type, the microtubules get attached side by side, forming a multi-layered structures which is in abbreviation called MLS, and the other part a single microtubule is there. And this unilateral type is phylogenetically significant because in advanced group of algae like Charophyta, this unilateral type can be seen, which is similar to land plants, which actually reminds us the Charophyta as the progenitor of land plants. In cellular biochemistry of algae, there are again variations due to varied chemical nature of algal cell wall, pigment composition and storage food products. If we discuss about the pigments of algae, we will find that chlorophyll A is the universal pigment present in all classes and divisions of algae. For this reason, the cyanobacteria having chlorophyll A is till date discussed by the algologist. The chlorophyll A is present inside the thylakoid membrane together with the other fat-soluble pigments like carotenoids and xanthophylls. The absorption spectra of chlorophyll varies at 663 nm and 430 nm. Besides chlorophyll A, chlorophyll B and chlorophyll C1, C2 are also present in different algal divisions, and chlorophyll D only in few members of Rhodophyceae. Chlorophyll B is present as major pigments in Chlorophyta and Euglenophyta, showing absorption maxima at 643 nm and 435 nm. Due to presence of major amount of chlorophyll B, the thalli of Euglenophyta and Chlorophyta are grass green in color. On the other hand, chlorophyll C1 and C2 are present in different algal divisions like Dinophyta, Cryptophyta, Primnaceophyta and Heteroconchophyta. They show their absorption maxima at different ranges of light spectrum like 440 nm, 452 nm and 583, 586, 635 and 634 nm. Here also, due to presence of chlorophyll C1 and C2 together with xanthophyll pigments, the plant body of Dinophyta, Cryptophyta and members of Heteroconchophyta are brown to golden brown in color. Chlorophyll D, already said, is present in some members of Rhodophyceae, showing the absorption maxima at 400 nm, 456 nm and 696 nm. Carotenoids are one of the most important pigments present in almost all members of algae, and absorption spectra of carotenoids at 450 nm. Carotenoids are present in different proportion. Alpha carotene and beta carotene are the main carotenoids present in algae. Besides this, lutein is also present. Oxidized product of carotenoids are xanthophylls, and xanthophylls are very important pigments of algae because they are brown to golden brown in color, and sometimes they are present in major amount in different groups like Heteroconchophyta, Phaeophyta and Bacillariophyta of these Heteroconchophyta groups, and Dinophyta and Cryptophyta. The fucoxanthin and the diatoxanthin and diadinoxanthin are brown in color and present in members of Phaeophyta and Bacillariophyta, giving the characteristics color of brown to golden brown to these thalli. Astaxanthin is one of the important xanthophyll present in Haematococcus, the member of Chlorophyceae. And these carotenoids and the xanthophylls are economically important also because they have high antioxidant properties. Now, besides these fat-soluble pigments, phycobiliproteins are present in Cyanophyta, Rhodophyta and Cryptophyta. These phycobiliproteins are water-soluble pigments and are associated with macromolecule of protein. The absorption spectra of these phycobiliproteins like C-phycocyanin, R-phycocyanin, C-phycoerythrin and allophycocyanin, they have absorption spectra at 565 nm, 615 nm and 652 nm. The algae can change the proportion of different amount of pigment according to the light intensity, and this property of algae is called chromatic adaptation. Like at scorching sunlight on mountains, the trentifolia turns deep red due to high amount of the carotenoids. When red algae grow in deep sea, their color is deep red due to the penetrance of only blue light under more than 200 meters depth. There are also variations in storage products of algae. If we consider the storage products of different groups of algae, we will find that there are different compounds like high molecular weight compounds. Among these compounds, the starch, Floridian starch and Myxophycean starch can be mentioned. These starch are the storage product of Chlorophyta, Euglenophyta and a few members of algae, which is actually alpha 1,4 linked glucans. Starch is composed of amylose and amylopectin and it turned blue color in iodine reaction. The starch are generally synthesized within the chloroplast at the site of pyrenoids. Floridian starch is the reserve food matter of the members of Rhodophyceae. It is also alpha 1,4 linked glucans, but here the amylopectin part is more in comparison to amylose. And it actually behaves like animal glycogen and it turned red-violet in color in iodine reaction. The Myxophycean starch is again proteinaceous in nature and present in members of cyanobacteria, which is also animal glycogen like compound. Now, beta 1,3 linked glucans or the laminarin, chrysolaminarin and paramylon are present in different members like Phaeophyceae, Bacillariophyceae and Euglenophyceae. This laminarin is actually the beta 1,3 linked glucans and present in members of Phaeophyceae, and chrysolaminarin is present in members of Diatom. Fructosans is 1,2 linked fructose present in different groups of algae. Now, low molecular weight compounds are also present in different groups like sugars and mannitol and glycerol. Sugars are low molecular weight compounds and present in members of Chlorophyta and Euglenophyta, and mannitol or the sugar alcohol is present in members of Phaeophyta, which actually controls the osmoregulation in marine environment. Glycerol is one of the important product of Dunaliella, which is again of economic importance. So, from this discussion, we can actually conclude that there are variation in morphology and in ultrastructure, in cellular chemistry of these well diversified groups of algae.